Hey guys, Bob here from Team Delta, and we're here to bring you growth hormones. And, it's kind of a long presentation, but I hope you guys just sit tight and enjoy it. I put in a lot of time editing this thing. So, happy holidays. Take care. Hey guys, Bob here. I'm actually doing myostatin, and as you can see, I have a bow tie, I got a half of a vest, and I have my pathway. Now, first off, what myostatin is, it's a negative muscle mass regulator, and as it sounds, you can actually find a way and go ahead and inhibit this protein complex, which is a complex to follistatin and some other stuff that probably don't have to get into. But if you find a way to go ahead and stop this from binding to its receptor, you can actually increase your muscle mass and look like Arnold Schwarzenegger. So I'm sure that has been in the market for many scientists for over the years. If you go to Google, you can actually type in Arnold Schwarzenegger mice, and you'll get some pretty buff mice there, and bulls, or if you're into that. All right, now down to the pathway. You can see here, this says L, and this says A, and you have this little arrow going up. The L is going to be your latent complex, and it's kind of like this protein complex of follistatin, myostatin, and you can actually get this cleaved. It's going to be cleaved by BMP, and you're going to get this active state right here, and it's going to bind to receptor 2, not receptor 1, but it's going to bind to receptor 2, and then move over, and it's going to start a phosphorylated cascade once it binds to receptor 2, or receptor 1 rather. And we're going to actually phosphorylate this protein called RSMAD, which is going to complex with SMAD, I know, and this is actually going to go into the nucleus and transcript your target gene. The gene in question is going to be something along the lines of P21, and of course that's going to stop cell growth just overall, and it's going to halt it completely. So if you have an overregulation of this myostatin, this can be really bad. People with cardiac infarctions have been noted to have high levels of myostatin, or just people with high levels of myostatin just have generally just less muscle mass, because you're not going to be able to build anything, really. All right, guys, and we have Celeste who's going to come up and talk about something. Good luck.
Gigantism is a disorder that is observed during childhood. What happens is in the hypothalamus, GHRH, or growth hormone release hormone, is produced and released to the anterior pituitary gland, and in the anterior pituitary gland, GH, or human growth hormone, is produced. This hormone is then released and sent to the liver, where the liver produces the IGF-1 factor, which is insulin-like growth factor 1. This hormone or factor then travels to the bones as well as muscles and produces growth during childhood. When too much growth hormone is released, it causes an excessive amount of IGF factors to be released and causes overgrown bones in children, which causes abnormally large children as far as height, muscle growth, as well as weight. Another thing that happens during childhood gigantism is sexual differentiation is triggered earlier because it is affected by bone age and not chronological age. So someone at the age of 7 would start their menses if they were a female, instead of starting menses during her 13th or 14th year of life. It also happens in males as well and causes a testicular growth at an early age instead of during the normal puberty age, which is about 13 or 15. This also occurs during adulthood, and the pathways are the same. There's a slight difference in the symptoms. This disorder is called acromegalia, and during this disorder, what it happens is the IGF factors are released and are sent to the limbs and the feet, and causes overgrown hands and overgrown feet. and it also causes a protruding jaw, and it affects the facial features of the individual, creating broader or more stern facial features. It also causes a larger tongue and prolongs the face and jaw bones. And this is only during the adulthood. So childhood is gigantism and the adulthood is acromegalia. Next up is Brandon with the production of red blood cells.
Hello, I'm Brandon, and today I will be demonstrating the functions under which the normal human body is able to generate and replace erythrocytes, also known as red blood cells, in the process known as erythropoiesis. Under normal conditions, the body synthesizes about 2.3 million erythrocytes per second. The process is regulated and monitored by compensating for small fluctuations in the blood oxygen content. When the body is exposed to conditions that induce hypoxia, such as exercise or high elevation, the flux is detected by the tubular epithelial cells and transmitted to adjacent paratubular endothelial cells within the renal cortex of the kidneys. The kidneys then undergo the process of synthesis and secretion of the glycoprotein hormone, erythropoietin. Also, the liver acts as a secondary producer of erythropoietin. The secreted erythropoietin then binds to specific receptors in the bone marrow, which once erythropoietin binds to receptors in the bone marrow, erythrocyte precursor cells begin a 7-stage developmental process, which all occur in the bone marrow. The initial stage is the transformation of a hemocytoblast, which is a pluripotent hematopoietic stem cell, into a common myeloid progenitor, which is a multipotent stem cell. The myeloid progenitor then develops into a unipotent stem cell, followed by further differentiation into a proerythroblast. The proerythroblast then develops into a erythroblast, which develops into a polychromatophilic normoblast. The polychromatophilic normoblast then develops into an orthochromatic normoblast, where the nucleus is expelled before becoming a reticulocyte. The reticulocyte is then released into circulation after about 1 to 2 days and becomes a mature red blood cell with a lifespan of around 120 days. After which, the cell usually undergoes the process known as apoptosis and releases the erythroproteins bound to its surface that will signal for the development of the new erythrocytes. In the condition known as hyperoxia, negative feedback will occur in which the erythropoietin production in the kidneys and the liver will be reduced, thereby decreasing erythroprotein production and inevitably, In the conditions of hyperoxia, negative feedback will occur in which erythropoietin production in the kidneys and liver will be reduced, thereby decreasing erythrocyte production. Synthetic forms of erythropoietin have also been developed to treat those with anemia resulting from chronic kidney disease.
Prolactin, and specifically pituitary prolactin, is the hormone that is primarily responsible for lactogenesis or milk synthesis. Prolactin in general is a very diverse hormone that has been shown to have not only various effects in humans such as angiogenesis, which is the growth of blood vessels and capillaries, as well as involved in lymphocyte differentiation and synthesis. It is important in the development also of the testes and ovaries, so sometimes it has gonadotropic effects, but it also has various effects in different animals. In fish, for example, it is responsible for the transportation of water and salt. Here I focus on the effects of prolactin, specifically from the pituitary, and what effects its activation has at post-parturition, so after birth. During pregnancy, lactogenesis is inhibited by the effects of progesterone, which levels are high during pregnancy, But after birth or post-parturition, when the levels of progesterone drop, the control of prolactin release from the pituitary is primarily under the influence of dopamine. Progesterone primarily inhibits milk synthesis in the alveolar epithelial cells, but I will concentrate on the continued levels of prolactin that is maintained post-parturition when progesterone is no longer present, and the inhibitory effects of this hormone are no longer present. Therefore, after birth, dopamine is the main inhibiting factor of the release of prolactin from the anterior pituitary, or adenohypophysis, and hence the main inhibitor of lactogenesis. As stated earlier, progesterone was the main inhibitor of lactogenesis, however, that is only during pregnancy, and that occurs primarily here at the alveolar cells, where prolactin would have already been released, and just its activation on the alveolar cells, so it halts lactogenesis that way, as to where dopamine inhibits its release from the anterior pituitary. And there goes my hypothalamus. Prolactin levels will only be sustained for about a week after birth. And after a week or so, the prolactin levels will fall off unless stimulated by some external stimuli. This external stimulus comes from when the newborn child is feeding or suckling. Mechanoreceptor, since lactogenesis process is both controlled by a hormone and a neuronal response, it is considered a neuroendocrine process. Mechanoreceptors on the breast send a signal to the hypothalamus, which then sends its signal via the hypophyseal portal system to release prolactin from the adenohypophysis, or the anterior pituitary. As stated earlier, dopamine is known to inhibit prolactin release, and so for simplicity, I will refer to it as prolactin inhibiting hormone, or PIH. There is also a prolactin releasing hormone, PRH, which is thought to be thyrotropin releasing hormone, or TRH. For simplicity here, I'll just refer to them as PIH or PRH. It is unclear, however, if it is the release of PRH from the pituitary, from the hypothalamus, or if it is the decreased release in PIH or dopamine that causes the release of prolactin from the lactotropes in the anterior pituitary. In any case, prolactin then travels to the alveolar epithelial cells, which are these little pink ones, which then causes the synthesis of milk, or lactogenesis. I should note that there is another hormone involved in the completion and successfulness of breastfeeding process, oxytocin, and those are the cells that it affects are these yellow cells, the myoepithelial cell, which is responsible in the letdown reflex, which allows the actual ejection of milk. Without both prolactin and oxytocin, successful breastfeeding cannot occur. However, oxytocin is not directly involved in the lactogenesis process. To recap, after parturition, prolactin levels are elevated or maintained via the stimulatory effect of the suckling, which an impulse travels to the hypothalamus, which then either through a reduction in PIH or an increase in PRH, or both, causes the release of prolactin from lactotropes in the anterior pituitary and milk. Okay. From there it travels to the alveolar epithelial cells, where it then activates lactogenesis and milk is synthesized and then secreted into the lumen of the alveolar lobule, which is then readily available for the next feeding or suckling of the next feeding of the next child. The next feeding of the child. That's perfect.
Platelet-derived growth factors exist as a family of positively charged homoinhibitor dimers of disulfide bonds, bonded A and B polypeptide chains. The mRNA sequence consists of 7 exon sequences, as you can see here. It consists of exon 1, which is the signal sequence, the central processing, the exon 2 and 3, which are the precursor sequence, exon 4 and 5, which will later be the mature protein of the PDGF, and exon number 6, which is the COH terminal sequence in exon 7, which is non-coding DNA, and extracellular release is dependent on the COOH exon 6 cleavage. Otherwise, the mature protein will reside within the cell. The mature protein consists of 8 cysteine residues. As you can see here, here's 8 of each one. and they form disulfide bridges between residues 1 and 4, and they can form either PDGF-AA homodimers, PDGF-AB heterodimers, or PDGF-BB homodimers. In order for these PDGF to elicit its response, it has to bind to homo or heterodimers of alpha and beta receptors. These alpha and beta receptors each consist of 5 immunoglobulin domains and a tyrosine kinase domains, which allows activation to form intracellular proteins to elicit its response. Binding causes cellular activation by autophosphorylation, as I said before, of the tyrosine kinases, and activation results in cell growth, chemotaxis, actin reorganization, and prevention of apoptosis. Some functions that PGDF-A homo or heterodimers can have is, one important one is embryogenesis or embryonic development, as you can see here. The beta receptor is known to elicit kidney development and blood vessel formation. The B receptor of either PGF-AB or PGF-BB is known to elicit regulation of myocardium synthesis and heart defects. And the alpha receptor, either PGF-AA or PGF-A, is known to elicit lung development and alveolar formation in the lung. And alpha receptor is responsible for cranial development and cranial formation. Another function it has is central nervous system development. It's known to elicit oligodendrocyte development and glial development and formation of the brain and spinal cord. PGF receptors are also known to play an important role in angiogenesis, which is the formation of cardiac muscle or blood vessel formation, and also to play a role in platelet aggregate formation. And also they play a role in tissue homeostasis, specifically maintaining interstitial fluid pressure, as you can see here. And also they are known to play a major role in wound healing. Some PGF diseases is the overactivity of PGF production has been linked to the progression of glioblastoma, which is the most common and most aggressive form of brain tumor, and sarcoma, which is the cancer development of tissues coming from such like the bone, fat, or muscle by autocrine stimulation of tumor cell growth, atherosclerosis, and myofibrosis, including lung fibrosis, kidney fibrosis, myofibrosis, and liver cirrhosis.
Hi, my name is Fiona, and I'm going to be speaking with you today about placental-lactogen. Placental-lactogen is an interesting hormone in that it is secreted during pregnancy by the placenta. Over the course of a healthy pregnancy, it is shown to increase by 16-fold in maternal circulation, and then drastically disappear from maternal blood following delivery. Placental-lactogen is a single-chain polypeptide composed of 191 amino acid residues, cross-linked through 2 disulfide bridges, which then fold into a secondary structure of 8 alpha helices. Compared with growth hormone, it is approximately 85% identical in its primary structure. Similarly, it also mimics the structure of the hormone prolactin. These similarities to growth hormone and prolactin allow for low affinity binding to the growth hormone receptors and high affinity binding to the prolactin receptors. This is providing insight into its role in embryonic growth and development. Placental-lactogen is also called chorionic somatomammotropin, and it's synthesized within the sensitiotrophoblastic epithelial cells of the chorionic villi of the placenta. This demonstrates direct evidence of its influence on pregnancy, as well as the female reproductive processes. Its primary role is lactogenic, and that it stimulates mammary gland development during pregnancy, but without causing milk secretion. It also has a luteotropic effect, in that it may supplement the action of choriogonadotropin on the corpus luteum of the ovary, signaling it to continue progesterone secretion, allowing for development of a successful pregnancy. Lastly, placental-lactogen has a maternal insulin inhibition function that stimulates growth-promoting action. It may alter maternal metabolism by countering the effects of maternal insulin, thus facilitating the availability of glucose, free amino acids, as well as vitamins and minerals to the growing fetus during the later stages of pregnancy. This function indirectly stimulates intrauterine growth necessary for the developing pregnancy, as well as directly stimulating fetal growth necessary for a healthy baby. As you can see, placental-lactogen has several important influences on human growth and development, both in the embryonic stages as well as maternal growth in support of the fetus. Thank you. Folks, thanks for hanging out with us on our journey of discovering the amazing capabilities of growth hormones. We hope you enjoyed meeting Billy, our paper doll, and we hope you learned something new. Be good out there. Bye-bye.